Biology 1107, presentation number 4, cells. We had mentioned once upon a time that the cell is a basic unit of life. It is the smallest part of life that demonstrates life's characteristics. So, a cell demonstrates hierarchical organization. A cell is made of cells, obviously. A cell has metabolism. A cell reproduces. A cell responds to the environment outside of it and to the environment inside of it. And finally, cells have DNA. So cells were first described by a fellow by the name of Robert Hooke way back in 1665. He was looking at wine corks through a rudimentary magnifier. And when he looked at the view of these wine corks, which were actually cut from plants, he observed teeny tiny little holes. Those teeny tiny little holes reminded him of prison cells, and therefore he called them cells. So Robert Hooke is credited with naming the cell and first identifying it, and you'll see his picture, and you'll see an inset of an illustration of cells from a cork and an illustration of the rudimentary microscope or magnifying instrument he was using. Now, a little bit later, I guess I would have to say about the same time, a fellow by the name of Antony van Leeuwenhoek, who was a Dutchman, was a hobbyist, and his hobby was grinding lenses, of all things. And as he was grinding lenses, he made up his own unique instruments that we could refer to as a microscope, although they don't really look much like a microscope. It's like a little handheld magnifier. And in using his handheld magnifier, he made observations of all kinds of living things. So his contributions were mostly descriptive, but he was looking at things that no one had ever seen before. I mean, he looked at pollen in plants. He looked at leaf tissues in plants. He even looked at human sperm. No one bothered asking him where he got it, but he observed human sperm and wrote all of these observations and documented them. So our study of cells begins with the identification of cells way back in the 17th century. So tell me more about this cell identity business, Knaus. You're telling me genetic identity has an influence on structural identity, has an influence on functional identity, has an influence on physiological identity. Well, what's a structural identity? Structural identity is the actual physical space that the cell occupies. For example, in the pancreas, I've got certain cells called beta cells that have a certain job to do. Those beta cells occupy a specific place in the pancreas. They have a structural identity by virtue of the physical space they occupy. But then again, they do something. Beta cells are critical in the production of insulin. And that is going to be the physiological identity of our beta cell. It occupies a space. It functions in the pancreas. More specifically, physiologically, it generates insulin, and it's because the genes code for that fact. A cell's identity is largely a consequence of which genes are turned on and which genes are turned off. If you think about it, a beta cell in the pancreas contains a copy of every single gene that makes you you. Now, obviously, a beta cell doesn't need to have genes turned on that code for eye color. A beta cell is not going to need genes turned on that code for neural development. You don't need nerves in your beta cell. You certainly don't need eye color in your beta cell. So those particular genes are going to be turned off. So yes, it's important to realize that every single cell in your body has a complete library of all of your genes, because every single cell in your body is derived from a zygote that started you off to begin with. But what makes those cells different has to do with which genes are turned on and which genes are turned off. So tell me more about this cell identity business, Knaus. You're telling me genetic identity has an influence on structural identity, has an influence on functional identity, has an influence on physiological identity. Well, what's a structural identity? Structural identity is the actual physical space that the cell occupies. For example, in the pancreas, I've got certain cells called beta cells that have a certain job to do. Those beta cells occupy a specific place in the pancreas. They have a structural identity by virtue of the physical space they occupy. But then again, they do something. Beta cells are critical in the production of insulin. And that is going to be the physiological identity of our beta cell. It occupies a space. It functions in the pancreas. More specifically, physiologically, it generates insulin. And it's because the genes code for that fact. A cell's identity is largely a consequence of which genes are turned on and which genes are turned off. If you think about it, a beta cell in the pancreas contains a copy of every single gene that makes you you. Now, obviously, a beta cell doesn't need to have genes turned on that code for eye color. A beta cell is not going to need genes turned on that code for neural development. You don't need nerves in your beta cell. You certainly don't need eye color in your beta cell. So those particular genes are going to be turned off. So yes, it's important to realize that every single cell in your body has a complete library of all of your genes, because every single cell in your body is derived from a zygote that started you off to begin with. But what makes those cells different has to do with which genes are turned on and which genes are turned off. Well, now we're going to make an observation about how cells are put together. It doesn't matter whether we're talking about a bacterial cell, a cell in a grasshopper, a cell in a pine tree, a single-celled organism living in a pond, or one of your cells. All cells have common structures. There are 4 structures found in all cells. Doesn't matter whether they're prokaryotes or they're eukaryotes, and you're going to need to know these. The first is called a cell membrane. The cell membrane is an interface between the cell and the environment around it. So if you're looking at this artist's rendition of a cell here, the outermost surface of that ball would be the cell membrane. The cell membrane allows things in, the cell membrane allows things out. In many cases, the cell membrane allows that cell to monitor what's going on outside of itself. Cell membrane goes by 2 other names. There's 2 synonyms that go along with cell membrane. Sometimes the cell membrane is called a plasmalemma, and sometimes it's called a plasma membrane. If you ever encounter those terms, they all mean the same thing. So what else do I find in common? All cells, once again, doesn't matter whether they're prokaryotes or they're eukaryotes, all cells contain a stuff inside of them that we call cytoplasm. Cytoplasm is the living content of the cell. It's the stuff inside the cell. Now, cytoplasm, as far as eukaryotic cells, includes 2 different things. If you take a look at our eukaryotic cell here as illustrated, I understand my eukaryotic cell does contain organelles, or tiny little organs. But those organelles are also enmeshed or interspersed in a matrix, in a semi-gelatinous material inside the cell. And all of the organelles are suspended in that gelatinous material. That gelatinous material gets a name. It's called cytosol. Cytosol. So the cytoplasm is cytosol plus organelles. It's all of the living content found inside side of that cell membrane. So the third thing I got down here is DNA, because every cell is going to have DNA. Every cell needs those instructions that are going to determine what its physical identity is, what its functional identity is, and what its physiological identity is. All cells have DNA. All cells have genes. All cells make proteins, and those proteins are encoded in those genes in that DNA. Finally, all cells, once again, doesn't matter if they're prokaryotes or eukaryotes, all cells are going to have ribosomes. Well, what's a ribosome, Dr. Canals? Well, ribosomes are tiny intracellular structures. And we mentioned this not too long ago. They're teeny tiny little, they look like teeny tiny little dots. And their job is to read the mRNA so you can make a protein. So obviously, if I got DNA present in a cell, I'm going to have ribosomes, because if I got DNA and I've got instructions for making a protein, ribosomes are my physical way of actually cranking out the protein. So all cells have four things in common. All cells have a cell membrane. All cells have cytoplasm. All cells have DNA. And all cells have ribosomes. All right, well, there's a really important practical consideration we need to cover at this point. We know that cells have function, they have physiology, they have genetic identity, they have physical identity. But there's one thing we got to deal with, and that is, why are cells so darn small? Even when I was a kid, I wondered, is it possible that there's a cell that's big enough for me to see with my naked eye? Is it possible that it might be a cell as big as a marble or a golf ball or even a basketball? Well, obviously the answer is no. I mean, I guess I could argue there are some cells that are big enough that I can see with the naked eye, but chances are probably not many of them. So why are cells so small? Well, we're going to find out that it has to do with the relationship between something called surface area and something called volume. If you take a look at a series of illustrations of three cells on this slide, a small cell on the left, a medium-sized cell in the middle, and a big cell on the right, you see they differ in terms of two distinct dimensions of measurement. They differ in terms of surface area, which is the amount of covering that each cell has, and that's the amount of interface and contact with the environment around it. And then there's the volume, which is the three-dimensional space that the cell occupies. Well, one of the reasons why cells are so small is they are attempting to maximize something called the surface area to volume ratio. So what's that, can ask? Well, that's the amount of surface area compared to the amount of volume. So on the next slide, we're going to try to understand the logical reason behind that. But right now, here's what I want you to notice. I want you to notice that in each of these cases, the cell has a certain surface area measurement and the cell has a certain volume measurement. So I'm looking at the really big cell on the right. The really big cell on the right, it's obviously going to have a significant amount of surface area. It has a surface area of 96 square micrometers. It has a volume of 64 cubic micrometers. Now hold that thought. There's a certain amount of surface area for every unit of volume we got. If I take a look at the next smaller cells, I see that the surface area is decidedly less, 24 square micrometers. But the volume is also substantially less, 8 cubic micrometers. However, if I was to compare the relative amounts, all I had to do is pull out my calculator. And let's see, I'm looking at the big cell. If I divide 96 by 64, I get 1.5. That means I've got 1.5 units of surface area per unit of volume. Now I'm going to turn my attention to the next smaller cell, and I said I've got what, a surface area of 24 divided by a volume of 8, and that gives me 3. I've got 3 units of surface area per unit of volume. Well, let's take a look at the smallest of the cells. The smallest of the cell looks like it's got a surface area of 6 square micrometers and a volume of 1 cubic micrometer. It's obviously a heck of a lot smaller, but let's go ahead and do that basic division. I'm going to take 6 micrometers squared, and I'm going to take 1 micrometer cubed. 6 divided by 1 equals 6. So you'll notice something. As I go from the largest cell to the middle-sized cell to the smallest cell, I'm actually increasing the relative amount of surface area, even though the cell's getting smaller. Well, let's take a look at the practical consideration. What does this mean to the cell? Because obviously there's some goal in mind. There's some about having a larger amount of surface area that is going to improve that cell's ability to live, survive, and do its thing. Well, okay, here's the logic. Surface area represents a supply of resources to the cell. Because remember, the surface area is the interface between the cell and the environment around it. So if the cell needs food, it comes in through the surface area. If a cell needs oxygen, it comes in through the surface area. Obviously, the more surface area I got, the more resources I can move into the cell. But now, on the other hand, you think about it, those resources got to be used. So where is the demand for those resources? Well, the demand for those resources is in the volume of the cell. So a big volume means a big demand for resources. A smaller volume means a smaller demand for those resources. So we can think about it as a supply and demand idea, that a cell is going to be really, really small because the smaller I get, I'm increasing the amount of surface area. I'm increasing the supply of resources for the amount of volume. Think about it another way. As a cell gets bigger, its surface area increases at a lesser rate. We saw that before. The teeny tiny cell's got a 6 micron squared surface area. The middle-sized cell has a 24 micron squared surface area. The biggest cell has 96 squared micron surface area, but compared that to the amount of volume, technically surface area is increasing at a lesser rate the bigger the cell gets. If it's increasing at a lesser rate, that means you've got less available supply, but a greater demand for those resources. So take a look at my little red print down there. The smallest cell features less demand but greater supply, and the largest cell features more demand and less supply. It features more demand because I got so much more volume but less supply because, comparatively speaking, the surface area is much less. So now there's a term to describe the relationship between volume and surface area, because I'm understanding as a cell gets bigger, surface area increases at a lesser rate. That means you got two variables here. I got a variable that is volume, and I got a variable that is surface area. And as the volume changes, the surface area changes at a different rate. If you got two variables changing at two different rates, that is described as an allometric relationship. So, take-home lesson, cells got to be small, because if you're small, you keep your volume down, but at the same time, you maximize your supply of resources. If you get too big, your volume gets so large that you've got too great a demand and not enough surface area to supply those resources. So if you're a cell, it's in your best interest to be small. By maximizing surface area, I maximize resources that will supply the volume with what I need to stay alive. Now, it wasn't too long ago basically way back in presentation number 1, we made a distinction between what are prokaryotic cells and eukaryotic cells. I want to revisit that topic, in part because I want to review what we already know, but I want to add one more distinguishing feature that exists between prokaryotic cells and eukaryotic cells. And it's a feature that we'll document a little bit later in a little bit more detail. But right now, you will remember prokaryotic cells are typically bacteria. They're prokaryotes. They have no membrane-bound nucleus. They have no membrane-bound organelles. As a rule, they're small and they're simple cells. But I want to take a look at the DNA inside the cell. Now, we know every single cell has DNA. Turns out that there's a basic difference between prokaryotic and eukaryotic cells in terms of how that DNA is packaged. If you take a look at our prokaryotic cell here, you will see what looks like a bunch of dark thread-like material dispersed throughout the middle of the cell. All right, this particular cell does not have a nucleus, but it does have a region, a place in the cell where that spaghetti-like DNA is found, and that's called the nucleoid. So I need you to make sure that you know that the word nucleoid corresponds to a region in a prokaryotic cell where we find the DNA. It sounds like nucleus, but it is not an organelle. It's a place. The other thing I want to notice is that in my illustration here, the DNA molecule looks like spaghetti. Now, if I was to unravel that molecule, it would be one big, huge, giant circle. So the DNA molecule in bacterial cells is technically a circular molecule, and I see the word simple here. And the idea is that this DNA molecule is not packaged with anything else. It's all by itself. Obviously, I can't really explain that until I talk about eukaryotic cells, where the DNA is in fact packaged along with proteins. Suffice it to say, at this point, prokaryotic cells have the typical prokaryotic characteristics we mentioned not too long ago. No nucleus, no organelles, small, simple. To that, we will add that the DNA exists as a simple circular molecule found in a region in the cell called the nucleoid. Now, on our next slide, what we're going to do is we're going to talk a little bit more about prokaryotic cell structure. We will go and visit eukaryotic cells in a bit. So what we need to do is kind of hold this thought, talk about the structures of prokaryotic cells, and then we'll visit eukaryotic cells. All right, well, we got the impression that prokaryotic cells aren't anywhere near as structurally complicated as eukaryotic cells, and that's true. But the fact of the matter is, just because it's not quite as complicated, it doesn't mean that prokaryotic cells don't have some structures we need to be aware of. So I see here an artist's representation of a bacterial cell, a prokaryotic cell, and it appears to be illustrated in a cutaway with a number of structures that are labeled. If you go ahead and Google bacterial cell structure or Google prokaryotic cell structure, you will get 101 versions of this same illustration. If you don't like this illustration, go find you another to look at. But this is what I need you to know about prokaryotic cell structure. Prokaryotic cells have a cell membrane. That's typical. I would expect that. It is the interface between the cell and the environment around that. But there's something else. Outside of that cell membrane is a non-living component of the cell called a cell wall. It's non-living. It has nothing to do with the life of the cell. It is composed of a carbohydrate that's characterized by amino acid cross chains. So it's a fairly strong structure, and it's composed of this compound called peptidoglycan. Now the name kind of gives me the clue. Peptido would imply amino acids bonded to one another by a peptide bond, and glycan would give me the impression of a carbohydrate. Peptidoglycan is the compound of which bacterial cell walls are typically made of. There are a few exceptions, but by and large, if it's a bacterial cell, it's It's going to have a cell wall, and that cell wall is going to be made of peptidoglycan. Now, if you take a look at our cutaway illustration of the bacterial cell, the plasma membrane or the cell membrane is labeled there in orange. The cell wall is illustrated in purple. And outside of that cell wall, there's one more, I would say, extracellular layer. And that extracellular layer is commonly called a capsule, but more officially, it's called a glycocalyx. In many cases, not in all, but in many cases, a bacterial cell has kind of a goopy, gelatinous layer surrounding it. And that goopy, gelatinous layer varies in terms of its physical consistency. That's why I use the words goopy and gelatinous, because they really imply two different things, but they apply to this layer that exists outside the cell wall. Sometimes it's very, very thin, as illustrated here, but other times it could be very, very thick. That thickness could be equivalent to the diameter of the cell. So the cell could be surrounded by this really, really thick layer of goopy stuff. And in some cases, that goopy stuff might be more rubbery or gelatinous. So the capsule is a layer of material that exists outside of the cell wall. And it varies in its thickness, and it varies in its consistency. Now, I guess I would have to ask the question, well, why would a bacterium have a capsule? What purpose would a glycocalyx provide for the bacterium? Well, I can think of two things. The first is that a glycocalyx can be an agent for that bacterium to attach to something. If you have ever gone to bed without brushing your teeth and you wake up the next day, you rub your tongue over your teeth and they feel really, really slimy. Now, the reason why your teeth feel really, really slimy is because your mouth is full of bacteria, whether you like it or not. And if you don't brush your teeth, those bacteria are going to start to reproduce, and they will reproduce and live quite happily all over your teeth surfaces. But the sliminess you feel are those bacteria, and they are literally glued down on each tooth surface by what amounts to being this glycocalyx, or this goopy outer layer. So it is a mechanism of adhesion. It might be in the bacterium's best interest not to be dislodged or to move. It want to stay in the same place. And a glycocalyx could be a really good agent for that. Now, the second one I can think of is that a glycocalyx can present a protective layer for the bacterium. I'm going to mention a bacterium, and you don't have to know the name. It's called Streptococcus pneumoniae. It is a teeny tiny little spherical bacterium that causes pneumonia. And Streptococcus pneumoniae comes in two different forms. There's what's called a smooth form, and there's what's called a rough form. Now, the smooth form is the bacterium has this really, really thick outer layer. The rough form doesn't have it. So, smooth form of Streptococcus pneumoniae has a glycocalyx. The rough form does not. But if I take a look at the smooth form versus the rough form, the smooth form is what I would call a virulent form of Streptococcus pneumoniae. It causes the disease. And it is very, very difficult for the body to fight that disease off. Our immune system is designed to deal with bacteria. However, if the bacterium has this really, really thick outer layer, it's going to be a tough row to hoe to get rid of it, because that bacterium has this thick outer protective layer. On the other hand, the other form of Streptococcus pneumoniae, the rough form, which does not have the glycocalyx, is non-virulent. It doesn't cause pneumonia, primarily from the standpoint that if this rough form bacterium shows up on your doorstep, your immune system is going to kill it and get rid of it before it ever has a chance to get established and cause the disease. So know what a glycocalyx is and know what benefits it confers upon the bacterium. What other structures do I have here, Canals? Well, let's talk about what's inside of the cell. Inside of the cell membrane is the content of the cell, which we call the cytoplasm. It is the living content of the cell. It's the volume of the cell. And in that cytoplasm, I'm going to find ribosomes. Now, I understand that ribosomes are found in every single cell because my understanding is ribosomes are my way of taking information and actually reading it and actually making a protein. So ribosomes are critical for protein synthesis. I see my region of the cell where the DNA is contained. That's the nucleoid. And now I'm going to go ahead and talk about two other external features of the cell. First are pili, and the second are going to be flagella. Pili are small hair-like extensions of the cell surface. You'll see them illustrated as little kind of reddish orange hairs here. And pili are really, really good in aiding the cell in adhering to surfaces. Remember I mentioned the bacteria and your teeth? Well, it is quite conceivable that not only are the bacteria going to take advantage of the glycocalyx to allow them to glue themselves down to the tooth surface, but pili will issue from the bacterial surface and aid in that. It's almost like if you were looking at, for example, an English ivy plant crawling up a brick wall. And if you look really, really carefully, you'll notice that the English ivy plant makes all these teeny tiny little roots all along the length of the stems. And those roots literally attach the English ivy to the brick wall. Same thing's happening here. Pili are an attachment function, an adhesion function that goes hand in hand with the glycocalyx. So what about this thing called a flagella canal? It looks like a whip. As a matter of fact, my understanding is at least in some cells, a flagellum is going to beat. So I suspect, Dr. Knaus, that flagellum has something to do with bacterial motility. Well, that's not a bad assessment. Yes, the flagellum is going to cause that bacterium to move. If I was a bacterium and I was living in somebody's tissue because I was infecting that tissue, I might want to be able to move. Not a whole lot, not much, but the fact of the matter is if I'm able to move into fresh, uninfected areas of tissue, I've got a continually fresh food supply. So that's the motivating factor for being a motile bacterium. But the thing that you're wrong about is that bacterium's flagellum does not whip around. It looks like a whip, but it isn't. So here's what I need you to know. I need you to know that a bacterial flagellum does result in motility, but the flagellum is solid protein. Being made of solid protein, that what appears to be a whip-like structure is solid. So how does it move the cell? Well, the way it actually moves the cell is you can think of that flagellum as functioning a little bit like a propeller or a prop or a corkscrew. Now those three structures, propeller, props, and corkscrews, they all share one thing, and that is they all rotate. So what I'm going to see is that flagellum, instead of whipping around, it's a solid structure and it's going to rotate where that flagellum meets the cell. And we can't see it, but there are things called motor proteins. These are proteins that will act when ATP is supplied. Just like a motor will turn when you supply electrical energy or when you supply gasoline. So in this case, this flagellum is going to rotate on account of these motor proteins. And as that flagellum rotates, it will either propel the bacterium through the medium in which it lives, or it will pull the bacterium through the material in which it lives, much like a corkscrew plows its way through a cork, or a prop forces a boat to plow through the ocean, or a propeller propels a plane through the air. So these are the structures of prokaryotic cells you need to be aware of. What we're going to do on the next slide is we're going to jump into eukaryotic cells, and we will pick up this idea of what makes eukaryotic cells different from prokaryotic cells, and we'll revisit this idea of DNA packaging. What do we know about eukaryotic cells? Well, eukaryotic cells are characteristic of animals, plants, fungi, and protists. We know that eukaryotic cells contain a membrane-bound nucleus. We know that eukaryotic cells contain membrane-bound organelles. We know that eukaryotic cells are larger. We know they are more complex. So what don't we not know? Well, what you don't know has to do with DNA and specifically DNA packaging. We already know that the DNA is going to be located in the nucleus, so that's a no-brainer. The nucleus is an organelle. It's a membrane-bound structure that contains all the DNA. It's like the genetic library found in the cell. The DNA molecules tend to be rather long. They contain an awful lot of encoded information, and in many eukaryotes, they are represented by multiple molecules, which we commonly refer to as chromosomes. Human beings have 46 chromosomes. 23 of them you got from mom, 23 you got from dad. Every single one of your cells has 46 chromosomes crammed into every single nucleus. That's a lot of DNA. That's a lot of genetic information crammed into a very, very small space. Now, it turns out in eukaryotes, instead of having a spaghetti-like circular thread-like molecule of DNA, what we're going to do is we're going to take that DNA and we're going to spool it. We're going to package it. If you take a look at an illustration at the bottom of this slide, you'll see DNA, and you'll see that DNA is a really, really long thread-like molecule that appears to be spooled around some structures there labeled as histones. What's a spool? Well, if you buy thread, you know that that thread is wrapped up on a little cylindrical thing. That's a spool. If you buy monofilament, it's packaged in the form of monofilament that's wrapped around some kind of structure, and that structure is called a spool. So in the cell, in the nucleus, in our chromosomes, we have these proteins, and these proteins are called histones. Histones are like little spools. And so what I'm going to do is I'm going to take this really, really long thread-like molecule, and instead of packaging it as a big tangled mess of threads, I am going to wrap it around these histones so I can package a heck of a lot of DNA in a small amount of space. And you'll see further on our illustration that those histones are where this DNA is spooled, and the combination of histones and DNA is what we're going to call chromatin. This particular illustration was probably taken from some French or German textbook, and you'll see chromatin is spelled with an E. Well, you can eliminate the E. It's chromatin, no E. But chromatin is DNA plus histones. It's DNA packaged with proteins. And you'll see that the ultimate packaging results in my chromosome, which means I've got a huge amount of a potentially entangled thread-like molecule organized into this thing called a chromosome. And in my heart of hearts, I got to understand, if I need to access any gene in that long, tangled mess of a molecule, all I need to know is find exactly which histone it is and unpackage it. It's kind of like going into a library. You go into a library, you got thousands of books. Where's the book you need? Well, you're not going to go through the library book by book by book trying to find a darn thing. You're going to go to the catalog of the library, find the book, find the number of the book, and find it in the stacks. Doesn't take much. So out of those thousands and thousands of books, you can find the one book you need relatively easily because you know exactly what shelf it's on. You know exactly what stack that shelf is in, and you got the little call number on the spine of the book. Same thing in eukaryotic cells. I mean, I've got thousands and thousands of genes to get a handle on. If I need that one gene because I need to make that one protein, and it might be critical, my life might depend on me being able to find that gene and access that information. All I need to do is find it, unpackage it, and read it, and make the protein. So big difference in terms of prokaryotes and eukaryotes, not just in terms of how big the cell is, how complicated the cell is, whether it's got a nucleus, not whether it's got an organelles or not, has a lot to do with DNA packaging. Eukaryotic cells are really complicated. As a matter of fact, take a look at this artist's illustration on the right. And you see the plant cell at the top and the animal cell at the bottom seem to be jam-packed full of different structures, and those different structures are going to have certain jobs to do. And that's what we're going to do in the next series of cell slides. We're going to cover each of these cell structures. So this slide is nothing more than our view of what's coming. We're going to talk about the structures of the eukaryotic cell, the cell membrane, the cell wall, the nucleus, the endomembrane system, metabolic organelles, other intracellular structures, the cytoskeleton, and motility structures. All of this represents a whole lot of jazz, a whole lot of structures that are critical to the life and operation of these eukaryotic cells. That's where we're headed. Now, I will tell you this. You could Google eukaryotic cell structure, and you'll come up with many, many labeled artist versions of these pictures right here. Some of them a lot more complicated, some of them less complicated. But as you go through this material, don't hesitate to Google the name of an organelle. Don't hesitate to Google the name of a structure. Don't hesitate to go to Google Images and look at that stuff, because to me, a picture is truly worth 1,000 words, and I would much rather look at a labeled picture than I would want to read a darn paragraph in a textbook. So keep that in mind, please, as we go through the next series of slides. Eukaryotic cells are really complicated. As a matter of fact, take a look at this artist's illustration on the right. And you see the plant cell at the top and the animal cell at the bottom seem to be jam-packed full of different structures, and those different structures are going to have certain jobs to do. And that's what we're going to do in the next series of cell slides. We're going to cover each of these cell structures. So this slide is nothing more than our view of what's coming. We're going to talk about the structures of the eukaryotic cell, the cell membrane, the cell wall, the nucleus, the endomembrane system, metabolic organelles, other intracellular structures, the cytoskeleton, and motility structures. All of this represents a whole lot of jazz, a whole lot of structures that are critical to the life and operation of these eukaryotic cells. That's where we're headed. Now, I will tell you this. You could Google eukaryotic cell structure, and you'll come up with many, many labeled artist versions of these pictures right here. Some of them a lot more complicated, some of them less complicated. But as you go through this material, don't hesitate to Google the name of an organelle. Don't hesitate to Google the name of a structure. Don't hesitate to go to Google Images and look at that stuff, because to me, a picture is truly worth 1,000 words, and I would much rather look at a labeled picture than I would want to read a darn paragraph in a textbook. So keep that in mind, please, as we go through the next series of slides. The first structure on our previous slide was a cell membrane, but we already know that all cells have a cell membrane, so it really doesn't bear any further discussion. The second bullet on our list of eukaryotic cell structures has to do with cell walls, and this does require some bit of focus. Now, you remember I mentioned that a cell wall in the prokaryotes was a non-living structure. The same thing is true in eukaryotes. Cell wall is a non-living structure. It's a rigid construction found external to the cell. The question is how does it get there? Well, the cell wall is a secreted structure. In other words, the chemical components of a cell wall are actually made inside the cell and are deposited to the outside of the cell. So when I take a look at eukaryotic cells, some cells do have cell walls and some do not. And if you do have a cell wall, it might be made of one thing or it might be made of another. Depends on what type of cell you are. So let's take a look at our list here. And we're not just going to limit ourselves to eukaryotic cells, but I guess we'll basically kind of take a bird's eye view of all cell walls, including the prokaryotes. Kingdom Animalia, to which we belong. Animals as a rule. Humans, cats, dogs, pigs, sheep, birds, reptiles, worms, mollusks, sponges, the whole range of animals. Animals do not have cell walls. It's the only kingdom in the entire realm of biology that does not have cell walls exclusively. You can hang your hat on that. It's an animal. It does not have cell walls. Now, in the kingdom Plantae, which are the plants, yes, we do have cell walls. Doesn't matter whether you're a tree, a pine tree, a magnolia, a vine, an herb, or maybe some moss plant or a fern. Your cells will have a cell wall, and that cell wall is constructed of something called cellulose, which we have seen before in our presentation on organic chemistry. Kingdom Protista. Now, these are single-celled organisms that, for the most part, are motile, and some protists do not have cell walls, but some do. It's one of the reasons why I have the word cellulose in brackets over there. Some protists don't have cell walls, but if you're a protist and you do have cell walls, there's a very good chance that those cell walls will be made of cellulose. But there's some exception to that. Now, let me make a quick mention about the kingdom Protista. It's something that we will document later, particularly if you get into Biology 1108. And that is this. The kingdom Protista is considered to be a grab bag of different evolutionary groups. In other words, the kingdom Protista does not represent one evolutionarily cohesive group. Members of the kingdom Protista are not necessarily evolutionarily related, which means when I look at the kingdom Protista, I see a grab bag of many, many different types of unrelated organisms. So yeah, I am going to find cell walls in some. Some won't have cell walls. Some, the cell walls might be made of cellulose, and some, the cell wall might be made of something else. If I take a look at the kingdom Protista, some of the protists are autotrophic. They can make their own food molecules. Some are not. There's a whole lot of variation in the kingdom Protista, and so in most cases, you're not going to really be able to say exclusively that the kingdom Protista has this distinct set of characteristics because they are such a diverse, unrelated group. But now let's take a look at our next kingdom here, the kingdom Fungi. This is a definitely cohesive group. All these organisms are evolutionarily related. They all have cell walls, and the cell walls are made of a nitrogen-containing compound called chitin. Interestingly enough, chitin is the same compound that is used in the construction of the exoskeleton of insects and the exoskeleton of crustaceans. If you eat shrimp, you know you got to peel the shrimp. What you're peeling off the shrimp is the exoskeleton. You know, if you're eating a crab, you got that hard outer shell. That's the exoskeleton. If you step on a cockroach and it makes this nasty, juicy, crunchy noise, the crunch is because of the exoskeleton. The exoskeleton in these animals is made of the same stuff that the cell walls of fungi are made of. Oh, I see the kingdom Eubacteria here. That is our single kingdom in the domain bacteria, and we already mentioned their cell walls are made of peptidoglycan. But then I've got this other kingdom here, the Archaebacteria, the only kingdom in the domain Archaea. And their cell walls will contain a compound called pseudomurine. Chances are I would never ask you for that name. However, at this point, taking a bird's eye view of all of life, I'm understanding the far majority of organisms, the far majority kingdoms, do have cell walls. And a cell wall provides protection and structural rigidity. And the cell wall can be made of different, very unique substances based on who has that cell wall. We're through talking about the external features of eukaryotic cells, the cell walls. Let's turn our attention to what's inside. And probably the most critical thing when we think about eukaryotic cells is this thing called the nucleus, because after all, the nucleus actually characterizes eukaryotic cells and distinguishes them from the prokaryotic cells. So what's up with this nucleus? Well, the nucleus is technically a genetic library. It is the organelle within the cell that contains these molecules of DNA that comprise the genetic code of the cell. The entire set of chromosomes. Each chromosome is equivalent to a single molecule of DNA, and all of the genes are found on those chromosomes. So in a human cell, for example, that nucleus is going to be jam-packed full of 46 really, really long chromosomes, each one full of different genes coding for all kinds of different proteins. So the nucleus, what does it look like? Well, for most part, the nucleus is a spherical structure inside the cell, and it's membrane bound. Now, not only is it surrounded by a single membrane, but it's surrounded by a double membrane. If you look carefully at the artist's representation here on this slide, or just go ahead and Google it. As a matter of fact, you can Google nucleus or Google nuclear envelope, and you'll see even electron micrographs, photographs taken with an electron microscope showing you these membranes. And there's two of them, and they're sandwiched very, very close together. It is a really, really critical distinguishing fact about nucleus. You don't see double membranes anywhere else. Now, this double membrane that surrounds the nucleus, also known as the nuclear envelope, is perforated. It's got little holes in it. And those little holes are called nuclear pores. And they are going to afford the movement of materials from the nucleus to the surrounding cytoplasm. Well, what kind of materials, Dr. Council? What I can think of is inside the nucleus, we will actually construct the parts of ribosomes. So we can export the constituent components of ribosomes from the nucleus to the cytoplasm, and that's going to require an opening. That's going to require a nuclear pore. And the other thing is that when we actually do take the information from the DNA, we're going to make a copy of it. And that copy is called mRNA. And the mRNA molecule is going to be exported from the nucleus to the surrounding cytoplasm. And once again, those nuclear pores will be my agent for getting that molecule from the nucleus to the surrounding cytoplasm. So inside the nucleus, I find DNA. I find chromosomes. I find what is called nucleoplasm. And that nucleoplasm is all of the stuff contained in the volume of the nucleus. Now, more specifically, some of that stuff, some of that nucleoplasm are DNA molecules packaged with histones. We mentioned this not too long ago. If we're going to package DNA and it's going to be a very, very long, very, very thin, very thread-like molecule, it might be in our best interest to package it by spooling it on a protein called a histone. So the combination of DNA spooled on histones is given the name chromatin. Now, to be really clear here, chromatin is a characteristic of eukaryotic cells, but you cannot refer to chromatin as being in a prokaryotic cell simply because in the DNA in a prokaryotic cell is not packaged with histones. So chromatin is exclusively a term used for eukaryotic nuclei only. Now, in the nucleus, not only will I find nucleoplasm and its component chromatin, I will find occasionally a small, dark body inside the nucleus, which scientists many, many years ago called a nucleolus, sometimes pronounced nucleolus, which literally means tiny nucleus. Now, back in the early 20th century, I guess it was, maybe even before, but early on, some scientists had a very clear notion that the nucleus had something to do with the control of the cell. They witnessed the fact that if the nucleus was present in the cell would demonstrate certain characteristics. If the nucleus was absent, that wouldn't happen. So we understand that the nucleus controls the cell. Now, early on, scientists thought about this little itty-bitty body, this itty-bitty little structure contained in the nucleus, and you'll see it in the picture there. You'll see the nucleus with its nuclear pores, and inside you will see this dark little body nestled right smack dab in the middle. Scientists were convinced that this thing was a small nucleus. As a matter of fact, they were convinced that if the nucleus essentially controls the activities of the cell, then this was the control center of the nucleus. It was like the CEO's office in a business. The CEO calls the shots. The CEO's office is located in administrative offices. The administrative offices basically tell the corporate area what to do, but the fact of the matter is the CIO, or CEO rather, is the seat of all ultimate power in the corporation. So, to make a long story short, they thought this was the nucleus. It turns out they were wrong. The nucleolus is not a tiny nucleus. The nucleolus actually has nothing to do with the operation of the nucleus itself. The nucleolus is a place inside the nucleus that cranks out the components of ribosomes. Now, we haven't talked about ribosomes in detail. We already mentioned, though, the fact that ribosomes are tiny intracellular structures that make protein. And their job is to read the copy of the DNA and assemble amino acids to make the proteins. This happens out in the cytoplasm. So ribosomes got to be made somewhere. They get made in the nucleolus, in the nucleus, and then those ribosome components, oftentimes called ribosome precursors, are exported from the nucleus to the cytoplasm, where they are assembled to make full-blown functional ribosomes. Outside the nucleus, encompassing the entire content of the cell, is this rather extensive network of membranes that are collectively referred to as the endomembrane system. Now, the membranes in this endomembrane system might be physically connected, and again, they might not. Let's take a look at the two components of what is the endomembrane system. The first component is probably the most extensive, and it's called the endoplasmic reticulum. And its descriptive name. Reticulum means network. Endoplasmic means within the cytoplasm. So this is a network of membranes that crisscrosses the cytoplasm. In many ways, what it does is it compartmentalizes the cytoplasm to make things a bit more efficient. There's two types of endoplasmic reticulum. One is called smooth endoplasmic reticulum, and the other is called rough endoplasmic reticulum. In the artist's illustration below, you will see the nucleus tucked up there in the upper left. And then you'll see what looks like a purplish, reddish set of membranes. That's the endoplasmic reticulum. The other component of the endomembrane system is the Golgi, which is going to be illustrated there in blue. More about that in a minute. Let's go back to endoplasmic reticulum. Now, if you wanted to, you could Google endoplasmic reticulum and click on images, and you will see pictures of smooth endoplasmic reticulum and rough endoplasmic reticulum. Most specifically, these are pictures that are taken with an electron microscope. You can actually see these things. Well, the difference between smooth and rough endoplasmic reticulum is the presence of ribosomes associated with rough endoplasmic reticulum. You might remember ribosomes are tiny intracellular structures. They are tiny intracellular structures that will be involved in making proteins. So I take a look at endoplasmic reticulum, and if I see little tiny dark granules associated with that membrane, it almost looks like the membrane is studded with these little granules. It's studded with these little ribosomes, and that is why we call it rough endoplasmic reticulum. As a matter of fact, you will see on the slide a little kind of bullet point over there on the right that says protein synthesis. The implication is, of course, if I got ribosomes, I will be making proteins. And that's what the rough endoplasmic reticulum does. Now, on the other hand, if I don't have those ribosomes present, my endoplasmic reticulum does not have that studded look. You don't see a membrane studded with small, dark granules. You see a smooth membrane, and because those ribosomes are not there, my smooth endoplasmic reticulum is going to be involved in something else. And for the most part, that something else is going to be metabolism. Now, in any event, whether I'm cranking out protein or whether I'm doing metabolism, I am still going to be making some kind of product. Metabolism oftentimes results in the manufacture of something. So my endoplasmic reticulum functionally is going to be involved in manufacturing raw materials. As a matter of fact, you could go ahead and write down raw materials to the right of endoplasmic reticulum over there somewhere where you might find room. But the idea is that if my cell is going to survive, if my cell is going to crank out finished products, either finished products it can use or finished products that something else can use in the body, that process is going to begin with the manufacture of raw materials. It's like if I thought about a factory building cars. The cars doesn't come out of thin air. It's because that factory accesses raw materials. The steel, the copper, the rubber, the glass, the plastic, those raw materials make it possible for us to build the car. And in much the same way in the cell, I'm going to have a place where I will manufacture raw materials that will be used for manufacturing finished product. And those raw materials will be constructed in the endoplasmic reticulum. Now, thing is, if I'm going to make raw materials in the endoplasmic reticulum, I'm probably going to want to export them. I need to get those materials out to the place where I'm going to do some manufacturing. So you will see the very last bullet point under endoplasmic reticulum, you will see the words makes transport vesicles. Transport vesicles are little tiny spherical structures. They're membrane-bound spheres, like little bubbles, and they contain those raw materials. Now, if you look at the artist's illustration, you will see that where those transport vesicles are headed, they leave the endoplasmic reticulum and they are headed towards this thing we're going to call the Golgi. So let's make the shift here. We've done talking about endoplasmic reticulum. Let's talk about the Golgi. The Golgi sometimes is called a Golgi apparatus. It is nothing more than a flattened stack of membranes. It looks an awful lot like a stack of pancakes. And I see that in the illustration. With a little bit of imagination, that blue thing there looks like a stack of pancakes. Work with me. You need to agree that looks like a stack of pancakes. Anyway, what's going to happen is those transport vesicles from the endoplasmic reticulum are going to travel from the endoplasmic reticulum to that side of the Golgi. Those transport vesicles will drop off their raw materials, and then those raw materials will be assembled into some finished product. And when I'm done assembling those raw materials into a finished product, it's time to ship that finished product out. Take a look at the bullet points under Golgi. Receives transport vesicles, i.e. from the endoplasmic reticulum. It assembles those raw materials into a finished product, and that finished product is going to be shipped out. And that finished product is shipped in the form of what we will call secretory vesicles. Comes from the word secretion. The idea is I'm making something, and that's how I'm going to ship that stuff away from the Golgi. So the question is, what happens with those products, Dr. S? Well, one of two things. Number one, those products could be used inside of the cell. It is not uncommon for a secretory vesicle to basically carry its materials elsewhere inside the cell where they can be used. On the other hand, some of those materials are going to be exported to the outside of the cell. As a matter of fact, looking at my artist's rendition here on the slide, I see secretory vesicles headed and fusing with the outside membrane of the cell. So you can think of the endomembrane system as a bunch of membranes inside the cell, and really the bottom line is, while they might be physically linked and sometimes they might not be, I don't see any physical connection between the Golgi and the endoplasmic reticulum there, but I do know in my heart of hearts that those membranes are connected by function. So I'm making raw materials in the endoplasmic reticulum, and I'm manufacturing those raw materials into a finished product in the Golgi. One more thing about a Golgi. The Golgi is essentially a structure that demonstrates directionality. Many scientists will say that a Golgi demonstrates polarity. Now, the word polarity for some of us implies positive and negative, but in this case, polarity indicates I've got one end that is distinct from the other. You can think about the Golgi as being a structure where I receive raw materials and then I produce finished products. Now, the raw materials come in on what's called the cis face of the Golgi. If I'm going to imply directionality, there's two words going to be involved here. One word is cis, which is going to imply substances coming in, and the other word is going to be trans, which implies substances going out. So my Golgi has directionality. It's polar. It demonstrates polarity. And I got transport vesicles coming in on the cis face of the Golgi. And then I've got finished products heading out on the trans face of the Golgi. We are now going to turn our attention to organelles that are involved in metabolism. Now, for plants, metabolism is going to involve making food molecules through photosynthesis and then breaking those food molecules to get ATP. It's kind of both are flip sides of the same coin. So let's take a look. Metabolic organelles in plants. We got these organelles called chloroplasts. And chloroplasts are diagnostic for plant cells. You don't have a plant cell unless you've got chloroplasts. And those chloroplasts are going to be engaged in a process called photosynthesis. If you take a look at the tree, you see the tree's got leaves. Follow the area, close-up view of the cells of the leaf, and you see those cells are jam-packed full of teeny, tiny little green things. Those are chloroplasts. And that's where photosynthesis takes place. So chloroplasts are going to be found in photosynthetic organisms, what we also might call photoautotrophs. And those would be plants, and those would be photosynthetic protists, organisms that we might commonly call algae, or might actually also be in a minority of bacteria. That's not on the bullet point there, but I really must tell you guys there are a few bacteria in this world that actually do photosynthesis. Not many of them. But the bottom line is if you're a plant, the bottom line, if you're an algae, the bottom line is if you are a chloroplast, your job is to absorb sunlight. And one of the reasons why you absorb sunlight is your chloroplast is full of photosynthetic pigments called chlorophylls. And it's the job of the chlorophyll to absorb that light energy. More about that in a future presentation. But right now, at this point, I need you to know that when a chloroplast does its photosynthesis thing, it's going to crank out food molecules. Those food molecules are like glucose. They're sugar molecules. They are considered to be collectively known as photosynthate. And that's nothing more than a highfalutin word for the products of photosynthesis. And so for many of us, when we see a plant, we understand that plant is taking in carbon dioxide, it's taking in water, it's absorbing sunlight, and it's cranking out glucose. Glucose is photosynthate. And that plant can then take that glucose and maybe store it as starch. Technically, if starch is storing glucose from photosynthesis, starch is photosynthate as well. So when you think about the word photosynthate, we're talking about the products of photosynthesis. Taking a look at my slide here, I see tree and leaves. I see chloroplasts in individual leaf cells. I see a close-up view of the chloroplast. The chloroplast is going to be using those photosynthetic pigments in the chloroplast to make glucose. And that glucose molecule contains stored energy. And you could draw an arrow from the sun, that shining sun. You can draw an arrow from that sun down to that glucose molecule, because ultimately it is the sun's energy that's going to be absorbed. It is the sun's energy that's going to find its way into that food molecule. Know that chloroplasts are involved in photosynthesis. So our next organelle is going to be the mitochondrion. So if chloroplasts are involved in making food molecules, well then mitochondria are going to be involved in breaking food molecules. Plants got to have both. A plant needs to make its food molecules, and then it's got to break those food molecules to extract the energy. So plants got to do double duty. They've got to make and they've got to break. They do the making through the agency of chloroplasts, which we just talked about, and they do the breaking through the agency of mitochondria. So let's take a look at mitochondria. I've got an illustration here of an animal cell, which is fine. We'll mention that in a little bit. But inside that animal cell, I see what looks like these little capsule-like organelles that seem to have an outer membrane and a highly folded inner membrane. Those are mitochondria. And there appears to be several of them here in the cell. So what does a mitochondrion do? What is it involved with? It says here that a mitochondria are involved in cellular respiration. As a rule, most cells have multiple mitochondria, and their job is to break down food molecules and make ATP. So let's take a look at our bullet points here. Mitochondria are involved in a process called cellular respiration. Yes, Dr. Canals, I remember cellular respiration is an event of catabolism. We're breaking something apart. Furthermore, Dr. Canals, cellular respiration is an event of oxidation. I'm breaking apart a food molecule and I'm extracting the electrons. And then finally, when I do extract those electrons, I will use the energy on those electrons to make this stuff, the cellular fuel called ATP. So all eukaryotes have mitochondria. Now, I started off this discussion by saying plants got them because plants have to break down their own food molecules, even though they make them on their own. But folks, animals got to have mitochondria too. Fungi got to have mitochondria. Any organism has to have mitochondria. It's got to take those food molecules and break them down. So yes, plants have chloroplasts and mitochondria, but animal cells have mitochondria. They don't need the chloroplasts because they eat their food molecules. Fungi, molds, mildews, and mushrooms, they've got mitochondria. Single-cell protists living in the pond have mitochondria. If you got food molecules, you need to break them apart. And that's where mitochondria come in. All eukaryotes, animals, plants, fungi, and protists, as I said before, a mitochondrion is going to be engaged in breaking food molecules. That's a case of catabolism. And when I do that process, I'm going to liberate those energized electrons. I am going to use that energy to make ATP. So at the bottom of the slide, I kind of simplistically show you a molecule of glucose and a big fat arrow pointing to ATP. And of course, the implication is if I got these food molecules, I'm going to break them apart and I can use that ATP to sustain life, to grow, and to reproduce. Make sure you're clear on what is a mitochondrion. Make sure you're clear on what is a chloroplast. Make sure you are clear on what they do. Make sure you're clear that photosynthesis is an event of anabolism. I'm making something. Photosynthesis is a case of reduction. I'm adding energized electrons to something. That something is a food molecule. On the other hand, cellular respiration is an event of catabolism. I'm breaking something down. I'm removing electrons from a food molecule. That's called oxidation. And I will use those electrons, at least I will use the energy on those electrons to make ATP or cellular fuel. Let's take a look at the cytoskeleton of a eukaryotic cell. The cytoskeleton, in many ways, is like the skeleton that you would find, for example, in our bodies. It provides us with structure. It provides us with shape. A big difference, though, is that while our skeleton consists of fixed components, what we call bones, in the actual cytoskeleton, that would be the skeleton of the cell, the components are somewhat variable, somewhat flexible, and somewhat transient. In other words, they can come and go. So let's take a look at the function of the cytoskeleton first. Now, it is a set of protein elements that provide the cell some structure and some shape. But you'll notice I've got the word non-static down there. Now, the word non-static means that these cytoskeletal elements, like I said, can be somewhat flexible. Not just flexible in terms of being able to bend or move, but also in terms of being constantly dismantled and reassembled. And so the entire cytoskeleton of the cell is somewhat in flux. I mean, it's not one static set of cytoskeletal elements like our skeleton consists of discrete, specific bones located in specific locations. The whole darn thing is very fluid and very unpredictable in many ways. Now, the cytoskeleton inside the cell not only does it provide its structure and shape, but it also anchors organelles and other functional molecules within the three-dimensional structure of the cell. If I need a mitochondrion, it might be located in a particular place. If I need an enzyme, it might be located in a particular place. On the other hand, I might want to move a chloroplast from point A to point B, and the cytoskeleton is going to do that job for me. brings me to the bullet point there that says motility. Now, what I've just mentioned, moving a chloroplast, is a form of motility. It's intracellular motility. By virtue of the fact that you can deconstruct and reconstruct these cytoskeletal elements, things can move inside the cell. As a matter of fact, if you think about the process of cell division, which we have not covered, but will involve the separation of and the movement of chromosomes into daughter cells during the event of cell division, those chromosomes move because of cytoskeletal elements. So the cytoskeleton will be involved in intracellular motility. But then we're going to see in a moment that these cytoskeletal elements will be important in the action of certain structures that actually move a cell through an environment. Hold that thought, we will get to there. Now, next thing we got to do, though, is identify these three proteinaceous components of the cytoskeleton. The three components are microtubules, which are the largest of the cytoskeletal elements. They are long, hollow tubes. And you'll see them illustrated at right. In the artist's representation there, those long, blue, hollow tubes, they're called microtubules, and they are composed of a protein that is conveniently called tubulin. The smallest of the cytoskeletal elements are microfilaments. Those are solid rods, and you'll see them towards the bottom of the artist's illustration in yellow. And those are composed of a protein called actin. Interestingly enough, that protein, actin, is the same protein that plays a very, very important role in the structure and the contraction of our muscle cells. Then, in between the microtubules and the microfilaments are the intermediate filaments, descriptive name telling me that I'm somewhere in between in size, but also these intermediate filaments can be somewhat variable in their shape. And those belong to a family of proteins called keratin, which incidentally is the same family of proteins that make up your hair and your fingernails and the toughness of your skin. Make sure you are aware of the three different components of the cytoskeleton by name and by size. Of those three, arguably probably the most important will be the microtubules, if only for the reason that they are absolutely critical in the movement of chromosomes during the event of cell division, but there will be a few other reasons why as well. Now we'll take a look at motility structures. Motility structures are going to be structures that are involved in moving the cell. And in another sense, they could also be instrumental in moving a medium of the environment that exists outside of the cell. We'll hold that thought. We'll see an illustration in just a moment. But I will mention this, and that is motility structures of the cell are essentially microtubular in their structure. Remember, I told you that arguably microtubules are the most important of the cytoskeletal elements. And I said, if only for the reason, the fact they move chromosomes during cell division. But if I look at these motility structures, flagella and cilia, which we'll mention in a moment, they have a microtubular structure. Now, if you take a look at the illustrations at the top right, you'll see a sperm cell and you see a flagellum, which is a long beating tail. Obviously, that long beating tail, by virtue of its beating action, is going to propel that sperm in a certain direction. But if you take a look at the photograph, which is actually a scanning electron micrograph of some cilia found on the surface of a cell in your sinuses, those cilia are going to beat, but they can't propel the cell because if you notice, the cell is actually anchored. Your sinus cells don't swim around, but what they do is they do have these long beating hairs that will beat and move and propel whatever stuff is in your nose, which is mucus. And mucus is like a flowing, thick, sticky layer that functions as a filter. picks up spores and pollen and dust and catches it and moves it out of the body. So in this case, instead of moving the cell, these motility structures are moving the medium outside of the cell. Now, I mentioned cilia and flagella. Cilia are short beating hairs. They generally occur in large numbers, and if you take a look at the artist's picture center right of the slide, you will see a slipper-shaped cell, which is a paramecium, a very common single-celled organism found in ponds and lakes. And you'll see that cell is covered in teeny tiny little hairs. Those are cilia. Flagella, on the other hand, are much longer, and they generally occur in fewer numbers. And so, to the left of the paramecium, you'll see an artist's representation of a sperm cell, showing you a flagellum. Now, flagella and cilia, even though they differ in terms of their length, and they differ in terms of the numbers in which they're found, they all have the same basic structure. And interestingly enough, it doesn't matter whether it's a flagellum in an animal sperm or a flagellum on a plant sperm. And yes, plants have sperm. It doesn't matter whether it's a cilium on the surface of a single-celled organism, like in the artist's picture there, or a cilium found in one of your cells in your upper respiratory tract. All of these structures, cilia and flagella, have a common internal structure, and that is an internal structure that is defined by microtubules arranged in a specific location and arrangement. You take a look at the right-hand side, bottom of the slide, you will see an artist's rendition, two cutaway views of a cilium. And on the far right, you will see an electron micrograph, a cross-section through cilia. For example, the short beating hairs that might be found on that paramecium. Now, in each of these cases, you will see that the cilium, or the flagellum, in cross-section consists of 9 pairs of microtubules arranged in a ring. And those 9 pairs surround 2 individual microtubules in the middle. That is commonly called a 9+2 structure. The 9+2 internal structure is the arrangement of microtubules found in flagella and in cilia. It doesn't matter how advanced the organism is. It doesn't matter how simple the organism is. Always that same particular arrangement. Let's take a look at some other microtubular structures. We're going to take a look at structures that actually have a role to play in cell division. Now, these structures, at least in animal cells, are called centrioles. Centrioles are unique to animal cells. They're not found in any other kingdom. They're not found in the fungi. They're not found in the plants. Only found in animal cells. And a centriole is actually a paired structure. It's 2 cylindrical arrangements of microtubules. You'll notice in the artist's kind of model reconstruction there, you'll see what appears to be 2 little cylinders. And those 2 little cylinders are composed of 9 sets of triplets of microtubules arranged in a cylinder. That constitutes a pair of centrioles, although together they are commonly also referred to as a centrosome. So just make sure that you know the word centrosome is equivalent to a pair of centrioles. Now, what centrioles do in an animal cell is establish the plane of cell division. If you take a look at my picture at far right, you will see what amounts to be an oval, which corresponds to the shape of an animal cell in process of dividing. The line that I've superimposed on that illustration is the plane of cell division. In other words, the chromosomes, which are incidentally illustrated on this picture as those dark, black, dense bodies are going to be pulling apart, one pulling towards the left and one pulling towards the right. Now, ultimately, when that is done, this elongated cell that's illustrated here will pinch into two halves. One half on the right being one daughter cell, one half on the left being the other daughter cell. In other words, that line is the plane of cell division. So how centrioles work is they always occur in pairs, but when a cell is about to divide, a single pair of centrioles will become two pairs, two centrosomes. And those centrosomes will migrate around the periphery of the cell at least to some point where one pair of centrioles winds up on the right-hand side of the cell and the other winds up on the left-hand side of the cell. As a matter of fact, if you look at this illustration, you'll see two poofy arrays of microtubules. Those microtubules are radiating from what looks like a couple of little orange dots. At least that's what they look like to me on the slide. And those little orange dots are those centrioles. So what I'm going to do is I'm going to take a step back. I'm going to take a bigger picture look at my animal cell. And that is this, is the combination of the centrioles and those radiating arrays of microtubules are collectively referred to as microtubular organizing centers. So for an animal cell, it is the site of what we will call the spindle fibers, which are the individual microtubules that will pull chromosomes during an event of cell division. I mean, I got to pull those chromosomes from the center of the cell to a pole of the cell, so I'm going to create a brand new daughter cell, and that's what these spindle fibers do. They're microtubules that hook onto these chromosomes and pull them in a certain direction. But now those microtubules, those spindle fibers, originate with this thing that we're going to call the microtubular organizing center, abbreviated as MTOC. The MTOC is a large structure that includes the centrosome or the centrioles at the middle. It's a pretty darn discrete structure in animal cells, as we see there. They're big and they're poofy looking, and it's nothing more than a big, huge array of microtubules are radiating out from where the centrosome is. And from that radiating array, I do see some spindle fibers that are attached to those chromosomes and will ultimately pull those chromosomes apart. But I will mention this. MTOCs will also be found in plant cells as well. Even though a plant cell does not demonstrate any structure that even remotely looks like a centriole, when plant cells divide, they will develop spindle fibers from these regions that we could call microtubular organizing centers, and those microtubules include spindle fibers which will connect to the animal cell chromosomes and pull them apart during an event of cell division. So microtubules are pretty darn important. They're important in cell division. They're important in the cytoskeleton of the cell. They're important in the structure of cilia and flagella. In talking about microtubules on the last couple of slides, I used the term spindle and spindle fibers. This might be a good time to take a closer look at those. Now, you remember I mentioned that spindle fibers are microtubules that will actually move chromosomes during an event of cell division. And we've not talked about cell division specifically, but if I'm going to create two new cells from one parent cell, then what I need to do is I need to take the chromosomes in the parent cell, I need to replicate them, and I need to pull one set to one pole of the cell and one set to the other pole of the cell, and then ultimately divide that cell into two new daughter cells, each daughter cell containing one set of chromosomes from the original parent. So the spindle is a three-dimensional array of microtubules. If you think of a cell, it's not a two-dimensional illustration. It is a three-dimensional structure. So if I'm looking at this illustration, what looks like an oval, I see those microtubules seem to originate at the poles of the cell. Those would be my microtubular organizing centers, and I'll see my chromosomes lined up in the middle. And I will see a series of microtubules that extend from the microtubular organizing centers to and through the chromosomes that are lined up at the center of the cell. So a spindle is an array of microtubules, a three-dimensional array, and it's associated with the process of cell division. And its purpose is to move chromosomes. What we're going to do is going to take a closer look at how that actually works. There are some really nifty, neat animations of how chromosomes are pulled across a microtubule. There is an inset that shows you that fact. You will see a big fat blue thing, which is a chromosome. You will see a microtubule below, and you'll see what looks like a little connector, what looks like a little protein that has rabbit ears. That is a protein called a kinesin. And what it's going to do is it's going to walk its way, follow the arrow across the surface of the microtubule, and as it does so, it's going to pull the chromosome along. You kind of think of it as like a tugboat in a harbor, because a tugboat in a harbor pulls a big ship in much the same way. But in this case, you think of that kinesin protein as like a little individual pulling a big rope connected to a big, huge, giant chromosome, and every step that individual takes, he pulls on the chromosome, and slowly, 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 that chromosome will move. So I need you to know about kinesin proteins. The word kinesin comes from the word kinetic, which means to move. So you think about kinesin proteins move something. They are going to be attached to the chromosome at one end, and they have two what appear to be legs on the other end, and they literally will walk along the length of the microtubule, pulling the chromosome all the while. And so during an event of cell division, you've got large numbers of these kinesins connected to large numbers of chromosomes. For example, in the case of a human cell, when it divides, I've got 46 pulling the chromosomes to the right-hand pole of the cell, and I got 46 pulling the other set to the left pole of the cell. And eventually, as those chromosomes move, I will get two new daughter cells. The chromosomes moving through the agency of these kinesin proteins interacting with those microtubules, which are also known as spindle fibers. On the next series of slides, we will be looking at what we will call intercellular junctions. These are places where cells come together. And they are generally characteristic of animal cells, but we will document a form of intracellular junction that occurs between plant cells. If you are at all curious and you want to see some additional illustrations, I would certainly encourage you to Google intracellular connections. You will see any number of artist representations and electron micrographs of these interconnections. So what we're going to do is we're going to document these on the next series of slides. The first one I want you to realize is something called a tight junction that exists between animal cells. You can think of it as a situation where you've got little protein rivets shown here in green. And those rivets connect one cell to the other. So you'll see two cell membranes here literally riveted together by these little green proteins. So we got a number of different places where the cell's actually connected to its neighbor, but there's spaces in between these rivets, which means there is some degree of movement between the cells afforded by those rivets. That's called a tight junction. Now, you'll remember in the tight junction we just looked at, we can actually move materials between the cells because the tight junction doesn't effectively close the connections between the two cells. In this case, we're going to see a junction that allows a gap between the cells. So materials can flow through the cytoplasm from one cell to another through what we are going to call gap junctions. A gap junction is nothing more than an arrangement of proteins in a ring that creates a tiny little channel. And what I notice is the proteins can open and the proteins can close and therefore afford movement of materials from one animal cell to another. A gap junction is our second type of animal cell interconnection. Our third type of intracellular connection is similar to gap junctions, except this is characteristic of plant cells, whereas gap junctions are characteristic of animal cells. We're going to talk about plasmodesmata. Plasmodesmata, or plasmodesma singular, is a cytoplasmic connection between two adjoining plant cells. Now, the big problem with plant cells is they got cell walls. So if we need to establish a cytoplasmic connection between two plant cells, not only do I need to establish the cytoplasmic connection, but that connection needs to traverse two cell walls. The cell wall belonging to the cell on the left and the cell wall belonging to the cell on the right. Plasmodesmata are a way that substances can travel through the cytoplasm continuously from one cell to another. If you take a look at the artist's illustration, you will see plasmodesma there, and you will see in the electron micrograph, those narrow strands are these cytoplasmic channels. Our final type of intracellular connection is a desmosome, and this is characteristic of animal cells. And transport is really not an issue with a desmosome. Now, you remember we mentioned tight junctions, which afforded movement of materials between the two adjoining cells. We understood there were gap junctions that afforded transport of materials between the two cells. In this case, a desmosome is a bunch of keratin fibers that connect two cells in a fixed manner so that the cells do not move with respect to one another. It's how a tissue maintains its physical stability. If you think about muscle cells as part of muscle tissue, and every single one of those muscle cells is contracting, and the whole muscle is contracting, what is keeping those cells from sliding apart from each other, or essentially contracting to the extent that they're not coming up with some cohesive contraction? Because we mentioned once upon a time that the cells of a tissue exhibit a degree of interdependency, and that couldn't be more true than in muscle cells, because muscle cells are going to need to be somehow connected to one another, because when all, and I mean all, of these muscle cells contract, the sum total of this contraction will result in the contraction of the muscle overall. And that begins with the fact that these muscle cells have to be connected to one another. Now, the proteins involved in creating a desmosome are actually members of the same family of proteins that are found in your hair, in your fingernails, and the same proteins that are found in those intermediate filaments in the cytoskeleton. So here you see two adjoining animal cells with their respective membranes, and you see those filamentous keratin molecules that are connecting the two. And as a consequence, the cell on the right will not slip or slide across the cell on the left because they are adhered to one another.